A fit body is a way to a happy life, and many of us these days hit the gym to get that perfect body. Have you ever been to a gym and after a strenuous exercise, your trainer asks you to take rest so that you can heal from the micro tears that take place in your muscles and tendons? Have you thought about how these tears heal? Well, there is a process of cell division that helps you in repairing this wear and tear, and that process of cell division is mitosis. Today, in this video, we are going to talk about mitosis. So what is mitosis? It's a kind of division that takes place in most of the cells in living organisms, except those cells which are responsible for the formation of gametes, that is units of sexual reproduction. So let's consider all the other cells. They are known as somatic cells. For example, the cells of your skin, the cells of your lung, the cells of your liver, the cells of your other organs that you can think of other than those which are forming gametes are all your somatic cells. And all these cells, when they have to increase in number or they have to repair or they have to perform some kind of growth, and therefore we need more cells, these extra cells will be formed by the process of mitotic cell division. Why? Because during mitosis, the daughter cells that are formed, they are exactly similar to the mother cell, both quantitatively and qualitatively. Meaning they are genetically alike, they are structurally alike, and they are functionally alike to each other as well as to their mother cell. That is why this kind of division is known as equational division. During mitosis, a cell which has either diploid or haploid number of chromosomes divide to give rise to two daughter cells with equal number of chromosomes. Just to remind you here, what are haploid and diploid number of chromosomes? The total amount of chromosome present in the sperm, egg, ovum, etc., that is known as haploid set. It is basically half the amount of chromosome that is present in a total cell of our body. We get half of this set from our father in the form of sperm nucleus and half of this set of chromosome from our mother in the form of egg nucleus that fuses to give rise to a complete set, that is 2n. Now this complete set of chromosomes is known as the diploid condition. So if your cells have a chromosome number of 46, we consider this to be equal to 2n. That is, for you, 2n is equal to 46. Now out of these 46 chromosomes, you have received 23 chromosomes from your father and 23 chromosomes from your mother. So what is the value of n for you? For the value of n for you is 23. So for you, the haploid condition, that is n, is equal to 23 that you see in case of sperm and egg, and the diploid condition, that is 2n, is equal to 46 that you see in any other cell of your body. So during mitosis, when either a cell containing 2n or n number of chromosomes divide, the number of chromosome in the daughter cell remains the same. For instance, if a cell divides to give rise to two daughter cells, the amount of chromosome remains 2n and 2n in both the daughter cells. You can obviously understand that there is an increase in amount of DNA, must have been an increase in amount of chromosome as well. For this, you can refer to the previous video, that is the video of cell cycle, where we have explained how this increase in amount of DNA has taken place. So what are the different stages of mitosis? After interphase, the M phase, that is the mitotic phase, can be divided into karyokinesis, that is division of the nucleus, and cytokinesis, that is division of the cytoplasm. Now karyokinesis in case of mitosis can be divided into four subphases: prophase, metaphase, anaphase, and telophase. While these phases are not absolutely demarcated from each other, they are overlapping, but still they have some specific characteristic features on the basis of which we can define or describe these stages. So let's see what are the events that are taking place in these different subphases of karyokinesis during mitosis. The first subphase of karyokinesis is prophase. Now, whenever we are talking about the different events taking place inside the cell or inside the nucleus during cell division, we will consider these following points. 1. The structure of the chromosome. 2. The location of the chromosome. 3. The location of the centrioles. 4. The condition of the nuclear membrane and the nucleolus. And 5. Any other special feature. If I just go through these points as far as prophase is concerned, during prophase we see that the chromatin reticulum, which till now was completely diffused in the nucleus as an entangled mass of thread, starts becoming prominent, and you see that instead of remaining as a thread, by the end of prophase, this thread-like entangled mass now becomes distinct and starts showing single individual thread-like chromatin. So from chromatin reticulum, now the reticular, that is the network, slowly starts forming individual chromatin fibers. Why does this happen? This happens because of the condensation of the chromatin fibers. What do we mean by condensation? By condensation, we mean that the chromatin fibers, which were thin and had a diameter of about 700 nanometers, starts 1. Dehydrating, that is losing water, and 2. Coiling. So it starts coiling and supercoiling till it attains a diameter of about 1400 nanometers. So you can understand that the diameter of the chromatin now becomes almost double, if not more. Therefore, they will become shorter, more compact, thicker, and therefore much better observable under the microscope. This condensation starts from prophase, but it goes on till the next phase as well. This makes the chromatin fibers, that is thin thread-like colored fibers, to change into rod-shaped thick structures which are known as chromosomes. So that was the structure. What is the location? All this while, the entire nuclear reticulum was equally distributed in the nucleus. Now, they slowly start moving towards the nuclear membrane. So they start migrating towards the nuclear membrane. What is the condition of the nuclear membrane and the nucleolus? The nuclear membrane and the nucleolus inside the cell starts degenerating or disintegrating. That is, the nuclear membrane starts breaking into small pieces so that the entire nucleus is dissolved and the nuclear membrane is gone, and the chromatin now can get distributed in the entire cytoplasm. The nucleolus also slowly dissolves, and by the end of prophase we see both the nuclear membrane and the nucleolus, they have completely disappeared. What happens to the centrioles? Now during the S phase of interphase, as we have seen in case of cell cycle, the centrioles had already duplicated. So one pair of centriole moves away from the other pair of centriole, and their movement is, they are pushed forward, or their movement takes place due to the growth of microtubular fibers in between them. Now these fibers are known as spindle fibers. Why? Because these spindle fibers are tapering towards the centriolar ends and wider at the center. By the time prophase is complete, the new centrioles, they have shifted towards the two poles, and in between them the spindle fibers are arranged. How many spindle fibers are formed? Well, that varies from cell to cell, but it is always more than the number of chromosomes that are present in the nucleus or in the cell. Last but not the least, What is any special characteristic of prophase? During prophase, we see that the cell is now getting completely ready and equipped to undergo a complete nuclear division. So those were the characteristic features of mitotic prophase. The next step of mitosis is metaphase. Now during this phase, the chromosomes have already condensed to their maximum capacity, and now from chromatin fibers they transform into chromosomes. These chromosomes are thick, smaller or shorter rod-shaped structures which look something like this. Now the chromosomes get attached to some spindle fiber originating from the centrioles, which are now located at the two poles. So what happens here? From the two poles, the centrioles give rise to proteinaceous microtubules. These proteinaceous microtubules are known as spindle fibers. Now the spindle fibers attach to the chromosome. Where in the chromosome? At the centromeric region. Now at this centromeric region, the chromosomes contain a proteinaceous scaffolding which is known as the kinetochore. What is a kinetochore? A kinetochore is a protein to which the microtubules of the spindle fibers can attach. So, during the start of metaphase, the spindle fibers which are forming from the two poles attach to the centromeres. Now, they start coming together so that the chromosome comes towards the equatorial plane. This is known as congression of the spindle fibers. So it's almost like the spindle fibers are holding the chromosomes with their centromere and bringing them to the center of the cell in the equatorial region and lining them up. This lined up region of the chromosome in the equatorial part of the cell looks like a patch or a band or a plate after staining the cell, and that is why this is known as the metaphasic plate or the equatorial plate. During mitosis, as you can see, each spindle fiber is attached to one chromosome. Now there are obviously spindle fibers which did not get hold of any chromosome. So if you have, let's say, 4 chromosomes in your cell, it is not essential that exactly 4 spindle fibers will be formed. There will obviously be more spindle fibers formed than the number of chromosomes. So there will be some spindle fibers which span from one pole to the other and do not have any chromosome attached to it. Such spindle fibers are known as continuous spindle fibers, whereas the ones which have chromosomes attached to it are known as chromosomal spindle fibers. During this arrangement, it has been seen that mostly the smaller chromosomes remain towards the center of the metaphasic plate, whereas the larger chromosome remain towards the periphery of the metaphasic plate. This arrangement of chromosomes during metaphase is a characteristic feature of metaphase which looks like this under the microscope in an onion root tip cell. It is important because when we study the next part, that is anaphase, we will see that the positioning of all the chromosomes in the equatorial region ensures that when they start moving towards the two opposite poles, their distribution or their division or their movement is equal towards the two opposite poles. I will explain this in a little more details when I come to that part. So as of now, as we understand that metaphase is a phase during which the chromosomes align themselves in the equatorial region. The chromosomes are in their highest level of condensation. Each chromosome consists of 2 chromatids and look like this, and of course that nuclear membrane, nucleolus, they have completely disappeared. The next phase of mitotic cell division is anaphase. Now if you remember, we had discussed this, that during mitosis, the mother cell, which is diploid in nature, contains 2n number of chromosomes, and the daughter cells, both the daughter cells are also diploid in nature and they contain 2n number of chromosomes each. So how does this number of chromosome double? In anaphase or during anaphase, enzymes like separase, they break down the centromeric region, thereby separating the two chromatids of the chromosome. Now each chromosome consisted of two chromatids. Now the centromere has been digested from the center, as a result of which the two chromatids separate. These two chromatids are known as sister chromatids when they are attached, right? Now when they separate, now they form daughter chromosomes. We know that each chromosome contains one chromatid. Each chromatid contains one DNA molecule. So when the two chromatids they stayed together, each chromosome had two chromatids, each chromatid with one DNA, which were exact copies of each other. Now I have separated them. So basically I have two chromosomes which are now referred to as daughter chromosomes. So how does this happen? During anaphase, separase divides the centromere from the center. So each chromosome divides into two daughter chromatids or daughter chromosomes, and they start moving towards the pole. Why do they move towards the pole? Very frequently we say that due to the shortening of the spindle fibers. Now spindle fibers are not contractile. Therefore, there is no contraction of the spindle fiber. We should say shortening. How does this shortening happen? If you consider the spindle fiber, they are made up of microtubular proteins. Now, from the end of the poles, that is towards the centriole, microtubular proteins slowly start separating or breaking out from the spindle fibers. As a result, the length of the spindle fiber starts getting shorter and shorter. What happens because of this? Because of this, the spindle fiber pulls the chromosomes, the daughter chromatids or the daughter chromosomes towards the two poles. So, if one daughter chromosome goes to one pole, its other pair goes to the opposite pole. In this way, we ensure that each and every chromosome is divided into two. One part goes to one pole and the other part goes to the opposite pole. So what have we done? If we had 4 chromosomes, we have divided to form 8 daughter chromatids. 4 goes to one pole, the other 4 goes to the opposite pole. Now we have equally distributed by keeping the chromosome number constant. Now this pull towards the poles makes the chromosomes attain their characteristic shape. So look at this. If you have a string and you put your hand exactly at the center of the string and pull it, what shape does it take? Yes, it takes a V shape. Similarly, if you take the string, put your finger slightly away from the center and pull it, what shape is it taking now? Okay, so it's an L-shaped structure. Take the string and put your finger almost towards the end of the string and pull it. It takes almost a curved J-shaped structure, and if you take the string and just pull it from its one end, it maintains that elongated structure which can be regarded as I. So the same thing happens in case of chromosomes. When these chromosomes are pulled towards the poles by the spindle fibers, they take their characteristic V, L, J and I shapes depending on if they are metacentric, submetacentric, acrocentric or telocentric respectively. After the anaphase is over, we see that chromosomal clusters have been formed at the two poles near the centrioles. This marks the end of anaphase and the beginning of the next phase, which is known as telophase. During telophase, which is the last phase of karyokinesis, the chromosomes undergo changes which are almost opposite of prophase. For example, during prophase we had seen that the chromosomes are condensing, that is coiling and becoming dehydrated, and therefore they are becoming shorter and forming chromosomes. In case of telophase, we see that the exact opposite happens. The chromosomes now de-condense, that is they uncoil and they take in water to go back to their thin thread-like chromatin form. Eventually, the nuclear membrane and the nucleolus reappear. So fragments of nuclear membrane were floating here and there in the cytoplasm. They come together to form nuclear membrane at the two poles surrounding the chromosomes. Nucleolus reappears, and as a result of which we see that two daughter nuclei have been formed at the two poles. The chromosomal spindles have already shortened and almost disappeared. The interzonal spindles are still there, but they will disappear soon as the cell moves on to cytokinesis. So this marks the end of karyokinesis in mitosis, where the nucleus has divided to form two daughter nuclei, and the daughter nuclei have equal number of chromosomes as the mother nucleus. Cytokinesis, or the division of the cytoplasm, in plant and animal cells take place differently due to the presence of the cell wall in plant cells. So in case of plants, the division of the cytoplasm or cytokinesis takes place by cell plate or phragmoplast formation, and in case of animals it takes place by cleavage furrow formation. In animal cells, during cytokinesis, we see that a small invagination of the flexible cell membrane appears from the region which was supposed to be the equatorial region, that is halfway in between the two daughter nuclei. Now this cleavage looks something like this, and it goes on increasing in its depth till it meets at the center, as a result of which the two cells, they separate from each other and they have formed two daughter cells. So it is simply because of the flexibility of the cell membrane that invagination can be formed, and the invagination, which is known as the cleavage furrow, meets at the center and therefore separates the two cells from each other. In plant cells, the division of the cytoplasm cannot take place by cleavage furrow. Why? Because the cell wall is rigid and therefore invagination cannot be formed from the cell wall. So what is the alternative? The alternative is that during cytokinesis, Golgi bodies, they come and deposit their content in between the two daughter nuclei. This content first is calcium pectate, which is the primary constituent of the middle lamella of the cell wall. This small plate-like region where the calcium pectate has been deposited is known as the phragmoplast. Golgi bodies are responsible for forming this. Now the phragmoplast slowly elongates towards the periphery of the cell till it meets the cell wall or the periphery of the cell. As a result, the cell has been divided into two daughter cells. Now Golgi body brings fresh layers of cellulose which is deposited inside or on the two sides of the middle lamella, and then primary cell wall or cellulose deposition can further take place. The point is that we still have to develop the cell membrane in the plant cell. So here, the endoplasmic reticulum, which forms proteins and lipid, after the formation of the cell wall, it comes and deposits the protein lipid, and you know that endoplasmic reticulum is responsible for membrane biogenesis, that is synthesis of biological membranes. So it synthesizes that part of the cell membrane that is missing. As a result, now the plant cell has been divided into two daughter cells. So what is the significance of mitosis? Mitosis primarily is a kind of cell division which contributes to growth, repair and maintenance in a living organism. Why? Because it is a type of cell division where the chromosome number remains the same and the daughter cells are exact replica of the mother cells. So if you have a cut or scratch anywhere and you need new skin cells, mitosis will set in. If you want to grow, therefore you want to increase your muscle bulk, the bulk of your bones, etc., new cells are needed. Again, mitosis will set in. And any kind of maintenance, like for example, when you lose one cell, every cell has a lifespan. So let's say after 100 days, if one cell dies, another cell will be formed again by mitosis. This is because you want to retain the characteristic of the cell that you are forming. However, there are other functions of mitosis also. The mitotic division ensures that the nuclear cytoplasmic ratio remains constant. You do not want the cell to have a huge cytoplasm as compared to a very small nucleus. So if the cell keeps on growing, so much so that the nucleus now is very small in compared to the volume of the cytoplasm, that nucleus might not be able to control the entire cytoplasm. Then you might want to break down that one large cell into two small cells with two nuclei controlling that volume of cytoplasm. Mitosis is also responsible for regeneration. Like when you lose a certain part of your body, you can't do that, but if the lizard loses its tail, then it can regenerate or reform its tail, and that is because of mitotic cell division. So as you can understand that most of the cells of our body undergo mitosis except the ones which are forming sperm and egg. Therefore, mitosis is extremely important, and without mitosis, we will not grow. There will be no maintenance, repair, healing, anything taking place in the body. However, if we lose control on mitosis, that can lead to formation of tumors or cancerous cells. So that was all about mitosis. Now let me ask you a question. Can you give me the name of a chemical and its source that is responsible for stopping cells at a certain stage of mitosis? Well, if you can, do write your answers in the comments below because I eagerly wait to read your answers. I hope you have enjoyed the video. Do hit the like button and share it with your friends if you did. If you haven't subscribed to our YouTube channel yet, hit the subscribe button right now and click on the notifications bell. Do check out the full courses on our website and Android app, Manocha Academy. Links are all given below. So let's stay connected together and let's keep learning.